50-year mystery solved. Researchers open black box of stem cell transplants. This is by the Wellcome Trust Sanger Institute. New research on the long-term behavior of transplanted stem cells in the body reveals how aging impacts stem cells survival and immune diversity, providing insights that could improve the safety and success of transplants. For the first time, scientists trace the long-term fate of stem cells decades after transplant, unveiling insights into a medical procedure that has remained a mystery for more than 50 years. Insights could pave the way for new strategies in donor selection and transplant success, potentially leading to safer, more effective transplants. Researchers from the Wellcome Sanger Institute and their collaborators at the University of Zurich were able to map the behavior of stem cells in recipients' bodies up to 3 decades post-transplant, providing the first ever glimpse into the long-term dynamics of these cells. The study, published in Nature and part funded by Cancer Research UK, reveals that transplants from older donors, which are often less successful, have 10 times fewer vital stem cells surviving the transplant process. Some of the surviving cells also lose the ability to produce a range of blood cells essential for a robust immune system. Over 1 million people worldwide are diagnosed with blood cancer each year, including cancers such as leukemia and lymphoma, which can stop a person's immune system from working properly. Stem cells transplants, also known as bone marrow transplants, are often the only curative treatment option for patients. The procedure replaces a patient's damaged blood cells with healthy stem cells from a donor, which then rebuilds the patient's entire blood and immune system. In the UK alone, over 2,000 people undergo this procedure each year. The cellular mystery of transplants. Despite being performed for over 50 years, many fundamental questions about how transplants work have remained unanswered. While they can be life-saving, outcomes vary widely, leaving many patients facing complications years later. Donor age has been known to impact success rates, but what happens at the cellular level following a transplant has been a black box until now. In this new study, researchers from the Wellcome Sanger Institute and the University of Zurich used advanced genome sequencing techniques to analyze blood samples from 10 donor-recipient sibling pairs up to 31 years post-transplant. By analyzing the mutations that occur throughout life in the donor and recipient stem cells, they could track how many stem cells had survived the transplant process and continue to produce new blood cells in the patient's body, an approach previously impossible. The team discovered that in transplants from younger donors, those in their 20s and 30s, about 30,000 stem cells survived long-term, compared to 1 in 3,000 in older donors. This drop could lead to reduced immunity and higher relapse risk, potentially explaining why younger donors often result in better outcomes. They also found that the transplant process ages the blood system in recipients by about 10 to 15 years compared to the matched donors, primarily due to lower stem cell diversity. Low mutation rates in transplanted stem cells. Surprisingly, despite the intense stress on the transplant process, stem cells gain few new genetic mutations as they rapidly divide to rebuild the patient's blood. This challenges previous assumptions about high mutation rates during transplantation. The study also identified other genetic factors, regardless of the donor's age, that help certain stem cells thrive following transplant. This range of genetic advantages could lead to the development of better treatments, making transplants safer and more effective for a wider range of patients. Dr. Michael Spencer Chapman, first author of the study at the Wellcome Sanger Institute, said, "When you receive a transplant, it's like giving your blood system a fresh start, but what actually happens to those stem cells? Until now, we could only introduce the cells and then just monitor the blood counts for signs of recovery. But in this study, we've traced decades of change in one single sample, revealing how some cell populations fall away while others dominate, shaping a patient's blood over time. It's exciting to understand this process in such detail." Dr. Markus Mann, senior author of the study, University of Zurich, said, "The research highlights that age is more than just a number. It's an important factor in transplant success. Although the hematopoietic stem cell system is amazingly stable over time, younger donors generally supply a larger and more diverse range of stem cells, which might be crucial for patients' long-term recovery. We hope to continue exploring other factors that affect long-term hematopoietic stem cell dynamics in order to fine-tune both donor selection as well as recipient bone marrow environments for optimal long-term stem cell function." Dr. Peter Campbell, senior author of the study at the Wellcome Sanger Institute, said, "The transplant process forces blood and immune cells through a type of genetic bottleneck. Our new approach allows us to investigate this bottleneck phenomenon more closely. We find that the bottleneck provides multiple different opportunities for some stem cells to thrive more than others in their new environment in the recipient. We believe it will be possible to find the genes responsible for enabling some stem cells to thrive better than others. These genes could then, in theory, be harnessed to improve the success of the transplant procedure," he said. And this is by the Wellcome Trust Sanger Institute on SciTech Daily. Please leave your comments and thank you for your support. Please support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.